This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 141, recorded on July 8th, 2011. Hi, everybody. I'm Vincent Racaniello, and this is TWiV, the podcast all about viruses. Huge crew today, starting here in my office in New York City, where I spy a Dickson Despommier.
Hey, Vince.
How are you, Dickson?
Doing well, Vince. Thank you.
We have the T-shirt look today, huh?
We do. This is Casual Friday. In fact, every Friday is Casual Friday.
Welcome back, Dickson. Good to have you.
Thanks. Pleasure to be here.
Might as well continue here in my office, right?
Yep.
Sitting right next to Dickson, our guest today, back for the second time.
That's right.
On TWiV, Matt Evans. Hey, Matt.
Hey, great to be here.
Two-time TWiver.
Two-time TWiver.
That's right. Well, there's been a long pause in between my TWiving. I think I was on episode 20, which probably had a listenership of about 2 logs less than now.
2 logs. Spoken like a scientist.
That's it.
You were, episode 20 talked about Hep C, and that's what you're back here for today.
Sure.
We should also point out that you were in the Department of Microbiology at Mount Sinai School of Medicine here in New York City, which is, of course, where I got my PhD.
That's right. You were Peter Plazy's first graduate student, right?
That's right.
Wow.
First graduate student.
You have an illustrious background, Vince.
No, I have a illuminated background. Also
David
joining us
Baltimore.
today, let's get them in on the conversation.
Here, here.
From Western Massachusetts, Alan Dove.
Good to be here.
It was so silent, we didn't know if you were still there.
Yeah, I was just biding my time, waiting to be introduced.
How are you, Alan?
Doing okay.
A little cloudy out there, I bet?
Yeah, cloudy and rainy, but it's supposed to clear up for the weekend.
Just like here. No sky visible, right, Dickson?
None.
And let's see what's happening in Florida, north central Florida.
I'll
where
bet it's different there.
we have Rich Condit. Hey, Rich.
Hi, everybody.
Oh, Rich.
Hey, Rich.
Hey, Rich?
It's totally overcast and raining.
Ooh.
Really?
Yep, and it had been raining for a while.
Oh,
I don't know what the
goodness.
temperature is. Probably in the mid-80s.
This is good for your drought, though. You have a drought going on.
We need this. We need this big time.
You really do.
Yep.
Is that raining all throughout the southwest, east, or just in your part of the neck of the woods?
It's not raining seriously enough to stop them from launching the shuttle.
Oh, that's
Right.
now? That's
It already
today?
happened.
That has happened.
Really?
Cool.
Wait till we get to the pics, dude.
Okay.
Excellent.
Nice. I didn't know it was today. It's the last one, right?
It's the last
Yep.
one.
Yeah.
It is the last.
Are they doing any phage experiments on this shuttle?
No, but UF has some plants on it.
Yep.
Really?
Marijuana plants?
I don't think so.
No, they're not.
Okay.
They're not going to
I
get
don't-
that high, Vince.
I don't. You know, if I was orbiting the Earth, that's the last thing I would need.
They're
Exactly.
Not, they're not genetically manipulable, right? You okay, Matt?
Besides,
Oh, no. I thought Alan made those jokes normally.
No, it's a gene. Actually, there's a gene for this.
I thought you were passing out there, Matt. I'm sorry. Okay, so we are here today to discuss viruses.
Yep.
Right?
You'd never know it.
No, you wouldn't. Not so far after 8 minutes. We have a paper today that is called A Genetically Humanized Mouse Model for Hep C Infection. So I thought we'd get Matt up here to do this with us because he is, of course, working on hepatitis C virus. He works on entry of the virus into cells. And in fact, Matt, when you were here last, we talked about your Cludin paper, right?
That's right. And actually, the last author of the paper we're going to talk about today was my co-first author on that paper, Alex Ploss.
He didn't found PLOS journals, did he?
No.
Sorry. I'm infected with Alan today.
yes.
So what's Ploss's position at Rockefeller?
He's an assistant professor in Charlie Rice's lab. So it's I think it's a research track position, and he has really his own empire growing in there. He has a number of postdocs that are really just turning out wonderful stuff.
Okay.
Okay, so this was published in Nature,
That's right.
and it's from PLOS, who is not of PLOS journals, and also on the paper, a lot of other people, but not Charlie Rice. Oh,
No, he
yes,
is.
there
He's
he
second
is.
to last.
Second
And
to last.
I know the first author as well, Marcus Dorner, is a postdoc in Alex's group in Charlie's lab, and it's it's really just a list of some amazing scientists here.
Okay. All right, so let's start on this paper. Tell us, what do we need to know, Matt?
Well, the goal of this paper was to basically construct a new small animal model to study the hepatitis C virus, HCV. There are animal models for this virus. The most famous one is chimps. That's the only animal that you can naturally infect with HCV. There are some other more difficult models, like mice bearing human liver transplants. Those are very difficult models to work with. You need a mouse that has a liver defect so it will accept a liver graft, and it also has to be severely immunocompromised. So you can't do things like vaccine studies in those kind of mice. Also, it's really hard to actually do large studies with these mice just because they're so fragile and hard to make in the first place. So in this paper, Alex and his colleagues tried to make a mouse model that could at least support entry of hepatitis C virus. We had previously worked on characterizing the factors that were required in a human hepatocyte to support entry of this virus. So we basically did a lot of screens to find the receptors that were available in hepatocytes but missing in other cell lines that were required for entry of this virus into a host cell. So Alex and I had found that occludin was important for this process, and previously Thomas van Haan and I worked together to find that claudin-1 was another factor required for this process in hepatocytes.
And then there are two other proteins as well, right?
That's
so
right.
Can I break in here just for a moment? I understand the liver structure to be that it has to first pass through sinusoids in order to get into the hepatic circulation.
Sure, the sinusoidal barrier in between the circulatory system and hepatocytes is not a really great barrier. It's fairly
It's loose
leaky.
junctions, you think?
And so things can often diffuse past that, I believe.
Got it.
And
Malaria can't. How's that?
And
It's too big, probably.
there's also possible mechanisms like transcytosis and things like that too,
okay.
but that's not fully worked out.
Okay.
Can you define sinusoids for me, please?
Sure. There are 5 different kinds of vessels in your body: arteries, arterioles, veins, venules, capillaries, and then there are sinusoids. So the sinusoids surround all of your endocrine glands. And if you want to look at the liver as a big endocrine gland because it produces all the serum proteins that you've got, you'll find sinusoids around islets of Langerhans, around the pituitary, around the adrenal glands. It's a mechanism that the body uses to get hormones and other compounds into the blood as fast as possible, into the venous return, into the heart, and then out into the body. So sinusoids are what drives the endocrine system.
Okay.
And the liver is loaded with it.
Okay.
All right, so Matt, the 4 proteins, occludin, claudin, what are the other 2?
So CD81, which is a tetraspanin, and scavenger receptor B1, which is an HDL receptor, have been shown to be essential for this process as well.
For entry of the virus.
For entry of the virus.
But we don't know if it's binding or entry or both,
No,
right?
that's really figuring out the mechanism by which these factors are being used is the hard part. And just because we know that there are 4 factors involved doesn't mean there aren't a dozen factors involved or more than that. It's just these are the ones that are most obviously important for the process. There are other factors that people are identifying all the time which appear to also regulate this process as well.
All right, so I'm coming from polio where we take one transmembrane protein, we put it in a mouse cell, and the virus gets in. So there's just one.
Right.
So why does this virus need 4? Or does polio need 4, but
Polio
the mouse
also,
ones work?
Yeah, polio probably also needs 4 or more factors.
Yeah. Okay.
And it's more that HCV is really, really specific for the cell types it infects. It's a little controversial, but definitely the primary target for HCV infection is the human hepatocyte, and any other cell type is poorly able to support entry, replication, assembly, all the events of the life cycle. And it's pretty much appears that this virus has just adapted itself really, really well to work in an hepatocyte, and that's made it really, really selective for the factors that are available there.
Okay.
And these 4 factors we've talked about so far are just entry requirements. We
That's
haven't
right.
gotten to ones that are internal replication requirements.
Right, and even that is still a wide open question. We know a few of those factors, but there's a long list we haven't identified, I'm sure.
And the 4 factors that we're talking about, they're all cell surface proteins, correct?
That's right. So CD81 and scavenger receptor are probably on the basolateral surface of hepatocytes, which is the circulatory system side. And Claudin-1 and occludin are tight junction proteins. And how those factors are being used by an incoming virion is also a pretty wide open question.
And are these proteins ordinarily in the human liver specific, or are they distributed elsewhere?
No,
as well?
that was one of the questions we asked a while ago was what factors are controlling the tropism of HCV cell entry? Only hepatocytes and human or chimp hepatocytes are able to support just entry of this virus into a host cell. And we found out the scavenger receptor and Claudin-1 likely control the tissue-specific tropism of the entry process, where they're expressed in a number of tissues in the body, but they only overlap at high levels in hepatocytes.
Okay.
And so that regulates why those cells are able to support this. Yes, Dickson?
My mind is turning like crazy right now as you're describing this, because the entry process for malarial parasites is co-paralleling what you're talking about right now, because once it gets past the sinusoids, it still has to get into an hepatocyte.
It's worse than that. It even requires a few of the same factors, too.
Ow!
So
here's my question, though. The envisioned model suggested this. Could you take an outer membrane or an outer protein capsid from a virus that can get into another kind of cell except for hepatocytes and wrap it around the HCV and get it to go into a different kind of cell, and then once it's in there, what would it do?
Well, you could try to redirect HCV. And probably the simplest but still really complicated way to do it is just replace the glycoproteins in HCV. But the problem is there are severe limitations to just replication.
So it's the cell type then?
It's multiple blocks at multiple levels. So it's really the HIV-1 problem all over again.
Okay.
Where even if you can get through one block in a mouse, you still have another dozen blocks you have to deal with.
And malaria too. That's just it gets into all bunches of cells, but it doesn't replicate except in hepatocytes.
Exactly.
Fascinating.
So in terms of what's controlling the species-specific tropism of HCV entry, Alex and I, when we discovered occludin, said, now we have a complement of factors that when we express the human versions of them in a mouse cell, we can actually get HCV entry for the first time. So we expressed all combinations of human and mouse versions of these factors, and we basically found that CD81 and occludin needed to be of the human origin to work. But you could use either mouse or human scavenger receptor or Claudin-1, and it didn't really matter. So that's nice that two of these factors, scavenger receptor B1 and Claudin-1, control the tissue tropism, and CD81 and occludin, although expressed in a huge number of tissues in the body, are more controlling the species tropism of this virus.
All right, so that's the basis for this paper, right?
Right, so now we could get entry into a mouse cell in tissue culture. Alex wanted to find out if you can get entry into a mouse.
Did you want to do this experiment, Matt?
I thought about doing this experiment. And Alex has actually a really great background to do this. He had animal experience, and he was really the right person to tackle this, especially with the resources he had on hand. Yeah, I think it would have been a difficult project to set up in a brand new lab that I was doing.
Weren't they using marmosets for this at one time also?
I think they've tried all kinds of animals for this.
There's a guy at the University of Chicago, I thought, this guy Reusner, or I thought I heard about that sometime during my graduate education. I guess not.
I'm not sure.
Okay.
All right, so how do you make mice with these proteins?
Well, I think the long-term way to do it would be to make a knock-in mouse or a transgenic mouse where the human versions of CD81 and occludin were replacing the mouse versions.
Right.
But that's not the fast way of doing it. So Alex cleverly decided to go for just raw overexpression of these factors in mouse hepatocytes specifically. So he used recombinant adenoviruses to express each one of the entry factors required that we knew about to be required. And he made huge quantities of these adenoviruses in tissue culture and hydrodynamically delivered those to mice. And what that means is that he basically did an IV tail vein injection of this adenovirus. The nice thing is that when you inject something into the circulatory system of a mouse, it tends to go to the liver anyways. And so these adenoviruses were already preferentially delivered there where they can infect hepatocytes. and express, at least transiently express, the recombinant genes that were cloned into them.
So it's just IV?
IV, tail vein injections.
Why do they call it hydrodynamic?
Well, I think the principle is you're actually injecting a large volume of solution
I see.
in a rapid way. So
Okay.
you're trying to basically force delivery of whatever your, typically plasmids or viruses. And this worked great. It worked reasonably well, where he looked at the percentage of hepatocytes, of the mouse hepatocytes that were expressing these factors. And he found, I think, any single factor was expressed in 60 to 90% of hepatocytes. Wow. So that was great. And then he put in progressive sets of different factors. And when he tried to express CD81 and include together, he could get basically 18 to 25% of the hepatocytes to express these factors. He also put in all 4 factors at one time, and he could get at least 5% of hepatocytes to express these 4 factors at once. So that was really pretty efficient.
That's pretty amazing, because you're talking about 4 hits there.
Exactly. Well, he's putting in, I think, 10 to the 11th particles. So
it just goes to show you it's hard to get a high MOI in an animal.
Sure.
In the cell culture, you could get all cells expressing all 4, right? You put in enough virus, put it in an animal, they're just way more cells.
So did he look at the Kupffer cells by any chance, just to see if most of the viral particles were being screened out by that sort of
I think he did look at what cells were expressing these factors, and it was mostly hepatocytes.
Right.
He didn't find a lot of other, I don't think he found other targets, and he could use that then just to he could purify hepatocytes to quantify the percentage of those cells that were expressing what he wants.
That's
at least.
Fantastic.
Right.
All right, next step.
So the next step would be to infect that mouse, right? And so we can make cell culture-derived hepatitis C virus, and this was a huge step that a bunch of labs took about 5 years ago. So he took this virus that he actually made in huge quantities as well, which is not very easy to do, and did tail vein injections of HCV cell culture virus, and was not able to demonstrate replication in these mice, and that was not a surprise.
Replication assayed how?
Well, in that case, these are, he used a reporter virus that would secrete a luciferase protein, and he could never see real expression of that luciferase gene. He also looked for the presence of HCV RNA in the mouse hepatocytes or the mouse livers, and he did find a slight boost in the presence of HCV RNA or the persistence of RNA in cells where he had delivered these entry factors, but it wasn't very long-term and it wasn't very convincing. So he again got clever and used a reporter system instead that allowed him just to look at entry of HCV.
So he says here that one of the problems could be that adenoviruses induce interferon.
Sure. So
Right.
I illustrated that replication is probably a defect, but also he did show that he was stimulating a number of ISGs or interferon-stimulated genes with just the adenovirus transduction, and some of them are already known to be very potent against HCV. So you're really stacking the deck against replication in this system.
So, question, if you take mouse hepatocytes and transfect in HCV RNA, can you get replication, or is there a replication block, that's on the molecular level?
No, there is a block. We can take just subgenomic HCV replicons. These are things that just replicate intracellularly. Drop those into mouse hepatocytes and barely see replication. If you use a drug-selectable replicon, so you can really visualize single events, you can get a few events per 10 million cells, but it's not very efficient, and it's unclear why you can select those events to begin with.
So you don't have to invoke an induction of innate immunity by adenovirus to account for the lack of replication.
No,
Just a non-permissive cell.
There are just 2 obvious blocks, and there could be many others as well.
Okay.
To why he didn't see good infection with cell culture virus in these.
So you
mice.
can't get past those blocks by selecting.
I think he's
variants.
doing that.
Yeah.
We're trying to do that. Probably a lot of labs are trying to do that. And if you could just make a virus that has adapted to a mouse cell to replicate or do other events, I think that would be a huge bonus.
And mice don't have their own version of this virus.
Not that we know of.
But dogs do, right?
Yeah, dogs are canine Hepasi virus.
Yes.
Except that virus probably is not a hepatitis virus.
That's right.
Yeah, and I've heard a lot of people accidentally say hepatitis virus on that name.
Yeah.
So,
I mean, when you're studying hepatitis C, you're studying liver cancer, yes?
you're studying a factor that leads to liver cancer
Exactly.
after a number of years.
Exactly. If you make a hybridoma out of mouse and human liver cells,
Right.
what happens to the virus?
It doesn't look like there's a trans-dominant inhibitor of HCV replication. In other
That's
words,
that's a really good question, Dickson. Good
Thank you,
experiment.
Richard. Thank
and it's
you,
a
Richard.
really hard experiment to do.
Really?
Yeah, these hybridoma. Well, if you fuse two cells, they're pretty sick to begin with, and then to do any kind of replication assay in that is challenging. But the reports that have done this say that you should be able to get replication in there. All right.
I mean, you could take a cell that's replicating and then fuse it and see whether or not it continues its replication.
No, that's a good idea. Yeah.
Another good idea.
Okay, so you were talking about the assay.
Right. So what Alex and his group did instead was use a transgenic mouse that had a genomic insertion of a firefly luciferase gene under control of a constitutive promoter, except this gene was inactive because of stop codons immediately upstream from it. What they did was they used a mouse that had LOXP sites flanking those stop codons so that if you express the Cre recombinase, those stop codons would be excised and the firefly luciferase cassette would be activated. So this is really just a way of genetically monitoring the presence of Cre recombinase. He then used a hepatitis C virus that he had engineered to express the Cre recombinase, and what theoretically what happened is that this virus would get into cells, express that recombinase, activate the firefly luciferase gene, and you would be able to assay this at any time. He was able to actually show that these mice, when transduced to express, with these adenoviruses, to express the human entry factors, were able to support activation of this firefly luciferase cassette when he infected them with this recombinant virus. This wasn't dependent on replication. So he could actually use hepatitis C viruses that had inactivating mutations in the polymerase gene, and he showed that he could get the same signal regardless of that. So it's not like he had a replication system here, it's an entry system, and that's important to point out. But it was reasonably robust, and I think I moved my mic, but yeah, it was reasonably robust and definitely permitted him to study the requirements for entry into mouse cells at this point, and in vivo as well.
So would you have predicted that the viral RNA coming in without replication would have been able to make enough Cre to activate the reporter?
I think it really depends on how much Cre you can produce, and I think it's predictable that if you made Cre at a certain level, one or two molecules should be able to switch the reporter over. Problem is how efficient would that be? He is actually delivering an awful lot of HCV virus to these as well, and he shows that it's dose dependent. And in fact, so he would assay this by basically doing bioimaging of live mice. Mm-hmm. So he would go through the adenovirus delivery, the hepatitis C virus delivery, and then after a certain amount of time, he would bioimage luciferase in these mice by injecting them with a substrate and using a very sensitive camera to visualize the production of light through their body.
So that's what this figure 1b is, right?
Right. So that's the first figure after a long line of supplemental figures that he put in. So this is a real tour de force in terms of effort.
Wow.
And so for the listeners out there, what you see in these images, and maybe we could even put this as an image for the episode, is a mouse with this, I guess it's a digitized colorized version of the signal, this blue and yellow and green stuff showing up over the liver.
Right, it's a heat map of the amount of light that's being produced over a certain background.
So that's about where the liver would be, Dickson?
That's about where the liver would be. Yep.
That's a big organ, obviously. It's the largest organ in the body.
But you see little spots elsewhere, but that's just background.
It's remarkable that you could do this in a life.
Yeah,
I think it's fantastic.
And the signal to noise is actually much better than we would expect.
It's amazing.
If you put in just human occludin and CD81, you get some signal. But in that one figure, he also shows that if you add more scavenger receptor and claudin-1, you generate a lot more signal. And it actually didn't matter if those were human or mouse, that you generated the same amount of signal in these mice. So that just reaffirms the message that we had in our last paper.
So he wasn't able to see a signal with just a virus that expressed luciferase, which would be
Right.
detected the same way. And yet, with the Cree activating the gene from
Yep.
the mouse, he was able to see a signal. So that must represent an amplification
Well, it's
somehow.
Right.
theoretically when you have this recombination event, you turn on luciferase and it should stay on. And so you need a few molecules of Cree to get that event, and then you ramp up expression of luciferase. And it's not directly related to the amount of replication you get then, because in
Okay.
this case he didn't get any. But he actually found that the luciferase signal was transient. And he thinks that that's because of an anti-vector immune reaction. And he even showed that if he depleted NK cells, they had a much better perpetuation of this luciferase signal. So that really fishes out what's going on in this system really well.
Right. So the system that the reporter system that they got the signal from is a catalytic system essentially. The virus only needs to get a little bit of its
Protein
genome
expression,
in.
yeah.
Right. A little bit of protein expression will give you a large amount of luciferase signal. Whereas if you have luciferase in the virus, you're dependent on the viral expression being high.
Absolutely. And then there's a number of reasons why this is really only happening in hepatocytes in these mice. One is that the hepatocytes are particularly good at taking up the adenoviruses, and probably HCV is being exposed to hepatocytes preferentially. But theoretically, this could be occurring in other tissue types because we're only looking at entry. So if you deliver the receptors to another tissue type in these mouse, you might be able to see that.
Yeah.
But you're still depending on good or reasonable translation off the input HCV genome. And that might actually work better in hepatocytes because of the presence of microRNAs that might stabilize the genome in those cells. And so there's a number of reasons why this is still only occurring in hepatocytes in this model.
Okay.
So from these, we can say that you just need CD81 and occludin.
They're essential for that, but they're made a lot better, or this process is a lot more efficient when you have more scavenger receptor and more Claudin-1.
Even the murine, right?
Right. So there's another experiment they did where they basically put in all combinations of mouse and human versions of these 4 factors, and it reproduces well the previous finding that human CD81 and human occludin are most important, in that you need both of those proteins available, and then either mouse or human Claudin-1 or scavenger receptor are sufficient. And it's possible here that the process isn't that efficient in normal mouse hepatocytes, maybe because there isn't enough Claudin-1 and scavenger receptor available to really drive this process efficiently.
What's the biological role in normal times of the scavenger receptor?
It's an HDL uptake receptor, a
HDL.
cholesterol
Okay.
uptake receptor.
Okay. But it will take up other things too, or not?
Well, it could be that HCV is actually using those processes specifically during entry.
No, I mean the scavenger receptor only takes up HDL.
I know that it has a preference for that. I'm not sure
Okay.
of other activities.
So at any rate, this all
Interesting.
is consistent with your previous results in cell culture,
Yeah, so
which is nice.
No, it's always nice to see that,
Yeah.
but it's this is a much
question.
bigger step than what we took before. And so he's gone ahead and used this system in a number of downstream experiments. One was, like I said, just to find out what the species contribution of each factor is. He threw in a couple mutants of CD81 and occludin that have been shown not to work well in cell culture, and he showed they also don't work well in this mouse model, or at least the entry activity is impaired. People have had a tough time studying scavenger receptor in cell culture because most cell lines express this protein. But what he had available was a scavenger receptor knockout mouse. So he showed that in those mice, entry was very inefficient, and he could enhance the entry of this virus with either mouse or human scavenger receptor to similar levels. So that just nails down that scavenger receptor is really important for this process. And then the big take home is, well, what can you do with this animal model? You want to be able to study hepatitis C virus biology.
But these mice don't get sick, obviously, right?
They don't get sick. I mean, they're barely they probably have an inflammatory response from. They already showed that.
And they don't make particles, right?
Nope, it's a single cycle transient assay to look at entry of this virus.
Okay.
And that's
Right.
it.
So you can study entry,
You
basically.
can study entry. So it is ripe for therapeutic development. The possibilities are you can use it to study the entry factor inhibitors or inhibitors of the entry process, and we are very interested in those for their therapeutic potential. He's also
Right, and they do a little miniature vaccine trial here, right?
That's right. So you can also finally study in a small animal model neutralizing antibody responses against the virus. And so people have tried to do that in chimps for a while, and it's just hard to do large-scale studies. But this would be an easier system, or at least a slightly easier system than a chimp to do antibody-mediated neutralization studies. So he did two things. He took these mice and he did passive immunization with antibodies against either one of the HCV glycoproteins E2 or one of the receptors CD81, and he showed that these antibodies just transiently administered to these mice could impair infection or entry in this system. So that's reassuring that it really shows that the signal he sees is a glycoprotein and entry factor dependent system. And then he used a vaccine that people have previously used in chimps and mice to elicit a neutralizing antibody response that could block infection in this system. So he used a live vaccinia virus expressing HCV structural proteins, or I think it's the core through the NS3 genes. This was developed by Fred Prince's group, where he showed that if you immunize chimps, you could provide some protection, better protection against homologous challenge of the virus, and some protection against heterologous challenge. So this HCV is a very diverse family of viruses, and it's hard to elicit a cross-neutralizing antibody response or cross-protective response. But at least he could demonstrate that this recombinant vaccinia virus could elicit some response in these animal systems. And so Alex was able to get this vaccinia virus, immunize mice with this virus, show that they generated antibodies against the E2 protein, and that those antibodies could protect those mice to a certain extent from challenge with HCV virus. He was also able to passively transfer those neutralizing antibodies into naive mice as well to really show that this response was productive.
Is this vaccinia vaccine being considered for use in humans?
I think it's, well, I'm not sure really what it's doing downstream now. A live vaccinia virus isn't the perfect vector for vaccines. You wouldn't be able to use it in any immunocompromised individual, that's for sure. But there is potential there, and it's generated some of the best responses we've seen.
In animal models, right?
In animal models, that's right.
Okay. So all the existing antivirals that we have couldn't be tested in this model because they go downstream of entry, right?
That's right. So the two antivirals that were just approved, actually, boceprevir and telaprevir, are NS3 protease inhibitors.
Mm-hmm.
So those would block replication of the virus intracellularly. So those shouldn't do anything in this situation.
Are there any in the pipeline that affect entry?
There is one scavenger receptor inhibitor that I think is in phase 1 right now. It's called from Itherix in California, and well, I don't know much about what's happened to it since some original discovery papers that came out a couple years
Mm-hmm.
ago.
Will HDL bind to hepatitis C virus?
So there is definitely a clear association of lipoproteins and HCV. If you looked in a patient's sera, you can find that the virus tends to migrate in a very low density fraction that is reminiscent of LDL or VLDL. So that was the first evidence for this. And now there's been really good work in cell culture to show that the virus and the apolipoproteins are part of the same complex. So this is a viral lipoprotein complex. Or viral lipo, whatever. So there's a clear dependence on entry of the virus on apolipoproteins, that this apolipoprotein E is definitely a component of the HCV virion, and if you block ApoE with certain antibodies, you can block entry of the virus. So there's probably some host factor dependent entry mechanisms for this virus, and maybe those are being conferred through receptors for ApoE on the cell surface, like LDLR, which is a long hypothesized entry factor for this virus.
Do you think it makes sense to now take these two proteins and make transgenics instead of delivering them by adenovirus?
I'm sure Alex is doing that
that?
right now. I think that this was just the easiest way to do it and show that the model had potential.
Right.
But if he's only getting 5% of the cells to express all 4 of these factors, theoretically this would be a much more efficient system if all of the hepatocytes were expressing the required factors at sufficient levels.
So this is proof of principle, really.
Absolutely, and I think
Right.
it's great proof of principle, and it provides a useful tool, but he's probably going to make it a lot better.
Yeah, and
So-
it tells you that it's worth making a transgenic mouse,
Yeah, I
which
don't.
would be a lot of effort to go to just in order to get the proof of principle.
I doubt they were waiting for that information, though. This Charlie Rice's group tends to just go for it.
Right. Okay.
So how do you see this being used down the road?
Well, either this or more optimal systems will be used definitely for vaccine studies. The important thing I didn't mention is this is the first small animal model for hepatitis C virus that has a competent immune system. Chimps aren't a small animal model, or nor are they really useful in large studies. But in this mouse, this is the first mouse that has an immune system that you can really study a vaccine response with. And so this is a great step in the right direction to studying neutralizing antibody responses.
Right. I guess the next thing to do would be to try to put in some of the factors that are involved in post-entry steps.
In replication, try to deal with that block next, basically.
Do we have any idea what any of those might be?
The most convincing is miR-122. It's a liver-specific microRNA that's involved in stabilizing the HCV genome. This was originally discovered by Peter Sarno, and I think he talked about this on an earlier TWiV. But it's not clear that has anything to do with the species restriction for replication of HCV, because the sequence is exactly the same in mice, and it's also expressed at high levels in hepatocytes. So you wouldn't suspect that adding more of that in a transgenic mouse would make a difference.
Yeah, that's a good point. It's the same exact siRNA, or mirror. Okay.
And then there's also potential that interferon responses are better in a mouse cell against HCV, but the factors that Michael Gale found, like IPS1, should be cleavable by the mouse version of IPS1, which is an RIG-I signaling molecule, is also cleavable by the HCV protease. So it's not like the HCV can't fight off the interferon response in a mouse cell either. So that's not the obvious way either. I think the best thing to do is to probably either adapt HCV to a mouse cell, select for mutants that are capable of using the factors available in those cells, or screen for human factors that can permit HCV replication in a mouse cell. And that would be akin to the same screen we did to find occludin in the first place.
How was that screen done?
We did a basic transcomplementation screen. We originally found occludin by doing a cDNA library screen, where we complemented mouse cells where we had already engineered all the factors that so we made a cDNA library out of a human hepatocyte cell line that supported entry, so it presumably encoded all the factors that were necessary for that process. And we screened for cDNAs from that library that could make a mouse cell able to support entry. And to make sure that we took into account all the necessary factors, Alex and I had previously transduced those cells to express human CD81, scavenger receptor, and claudin-1. And it was in that cell that we had found that human occludin could make those cells able to support HCV entry.
And so what was your endpoint assay then?
Well, we used lentiviral pseudoparticles that were drug selectable and that we had put the HCV glycoproteins on top of. And so these virions had the tropism of conferred by the HCV glycoproteins.
Okay.
And so if they infected a cell, they could confer drug resistance, so we could select that population of cells that became infected.
Okay, so it was strictly an entry assay.
It was strictly an entry assay, and it was a really difficult entry assay because there was some inherent non-glycoprotein dependent entry of these pseudoparticles into host cells. So we had to use an assay that was actually developed by Paul Benash and Theodore Hatziano at the Aaron Diamond, where we could cycle cDNAs through multiple rounds of selection. We could actually transduce cells to express cDNAs, do our HCV pseudoparticle selection, then repackage the cDNAs within that population of cells and do multiple rounds of screening. And it was a terrific assay, actually.
So if you were going to screen for factors that supported replication, your readout's got to be different. You have to be scoring for actual RNA replication.
Right, and we have all the tools for that.
Okay.
We could use the same cDNA library technology, but in this case, instead of lentiviral pseudoparticles, we could actually just transfect them with HCV subgenomic replicons that encode drug selection cassettes, and we could select those cells based on just sustained replication of those replicons.
Okay.
This is something we really probably should get going on, actually.
More hands.
Yeah,
Or money.
I could use the money.
All that's in TWiV 20 for those who want to hear it in even more detail.
The screen, right.
A long time ago. I have one other approach for you, and that is to find a naturally occurring hep C of mice.
Sure. I
Right.
think it's the discovery of a canine hepacivirus makes it more likely that we can find a wider range of these types of viruses.
You just need to go and look. It's probably out there.
Go out there and check out all the mice. Find the ones with the yellow eyes.
Weren't you going to do a mouse virus study, Vince?
Yeah, it's in the works. I am looking for a couple of weeks where I'm here so I can trap them.
Well, call me first when you find the hepatitis virus.
Well, I'm going to take the livers out
Just watch out for Hanta.
because it's very easy to cut the liver out while you have the chest open and freeze them in case anyone wants to look at it, because I'm not going to do it. But
There are mouse hepatitis viruses, right? But they
those are
aren't,
coronaviruses, yeah.
right.
Those are different types of viruses.
Yeah.
It's called viruses are called hepatitis viruses based on their the disease
Based on
they cause.
pathogenesis.
Right.
Yeah.
Right.
All right, I see there's a note here, and so I will read all the authors off here of this paper. Marcus Dorner, Joshua Horwitz, Justin Robbins, Walter Barry, Kian Feng, Kathy Mu, Christopher Jones, John Scoggins, Maria Catanese, Dennis Burton, Manson Law, Charlie Rice, and Alexander Plas Pathogens. I mean Plas.
I think it's spelled differently.
Yes, it is. Good job. It's a nice paper.
It's
A
great.
lot
Yeah.
of folks working real hard.
A lot of data. But here's my objection, and it's not to the science. It's to putting supplementary data somewhere else other than in the paper.
Please.
If it's important, put it in the paper. I know this is a effort by the journals to conserve space.
Right.
But it doesn't do the science any justice to have to go and you're reading a paper. Let's say you print it out and you bring it on the train with you. Supplementary figures 1, 2, 3, 4, 5 before I can even get to figure 1 in the paper, and they're on online only. So
Right.
I object to this. Alan, I understand why they're doing
Well,
it.
you
but-
can print the
Well,
supplementals.
and I understand why they're doing it, but I also object.
Good.
Not only for that reason, but also, and I've harped on this before, the archiving problem.
Yes.
You know, there is, at least in the case of Nature, there is a paper copy of this somewhere. Some of these other journals, especially the open access ones, there's no paper copy of anything in existence. But there's a paper copy of this paper somewhere that's going to sit in a library, and a century from now, somebody will be able to pull this out and say, hey, this was the original paper where they did this. And they're going to look at it and say, isn't it cool? Wait a second, supplementary figure 1.
Whoops.
Where would we find that?
Exactly.
And it's going to be in some obscure format that nobody has heard of anymore called a PDF, and nobody's going to know how to read it.
No, it's out beyond the Kuiper belt. It's definitely
Right.
How much of this is a result of the review process, where in order to satisfy criticisms
Yeah,
of
a
the
lot.
reviewer, you add more
I've done a lot.
supplementary data and it kind of accumulates?
All right.
Well, it's sort of self-fulfilling. The existence of supplementary data feeds the existence of supplementary data because reviewers know that they can demand more data be included because it can just be shoved into the supplementary stuff, and that raises the bar, and that's good in a way. People are insisting on better science, but it's bad for the reasons we outlined. It shouldn't be shoved off into supplementary digitized files.
Maybe this will be part of this new open access journal policy that we were talking about last week. They said they wanted to limit the number of reviews. Maybe they'll address the supplementary data issue as well.
Yeah, maybe.
Well, if you don't have a print version, you can have all the data in the paper, right?
Sure.
Yeah.
Now, even the PLOS journals have a printed version. They have a PDF version anyway, so you can't have all the supplementary data there. But I think if you're all online, you might as well just put it there and have a link to it. Alan, someday there's not going to be paper, I'm sure.
Yeah, I know, and I'd be fine with that if somebody had figured out how to archive digital data, but nobody has yet.
Right.
empirically.
Well, the problem is,
and I think
yeah.
unfortunately we've leapt into. I'm all for digitization. I love it, being able to grab papers instantly, and it's wonderful. It's got a whole lot of benefits, but jumping into that before we've figured out how we're going to archive the scientific heritage of mankind is really premature. And I think I think there needs to be a little more attention to storing this stuff for the next 1,000 years.
Yeah, it may sound like I'm being inconvenienced by having to look somewhere else, but in fact,
Oh no, I'm completely sympathetic with that.
Yeah, sure.
Absolutely.
I just want it in one place because even a reviewer, it makes it difficult to review the paper.
Yeah.
Okay. All right. Anything else, Matt, that we should touch on before we leave?
No, I think we covered most of the points.
All right.
Good paper.
Let's do some email. First one is from Lance.
This is the same Lance that we read from last week, who was talking about vaccination against hepatitis B.
Yeah, that's right. We have quite a few emails from Lance, which is fine. Yeah. Many thanks to all of you for such an excellent podcast. I think you all do a really fantastic job. As an ID physician and postdoc working on flavivirus immunology, in my case, you are preaching to the converted. And as I am already an enthusiast for all things virological, but I hope this podcast grows in profile. As it grows in profile, you can reach an increasing number of people as you are so easy to understand and so good at explaining things, and I am telling as many people as I can about it. I'm really learning a lot. I have just been listening to TWiV 129, and Rich, I think, was talking about reverse genetics of viruses. I think one group of viruses for which this has been particularly difficult is the flaviviruses and Japanese encephalitis virus in particular, which still doesn't have a working cDNA infectious clone despite many attempts and several reports in the literature. The clones seem to be either very unstable or toxic to the cells used to grow them. That said, there is an interesting report in last month's Journal of Virology where the authors show this is because of E. coli promoter sequences in the Japanese encephalitis virus and dengue 2 genomes, which result in viral protein expression and toxicity during the cloning process.
Bummer. Get rid of those.
Yeah, and those are just random promoters. They're not there on purpose. It's just
Right.
you don't need much to have an E. coli promoter.
Right.
Yeah, this is common that you can get expression in E. coli. It's happened with some other full-length clones as well, and there are a variety of ways around it.
Yeah, I've talked to a few people who ended up building their constructs backwards and doing other things like that to try and avoid the promoter.
Also relating to Japanese encephalitis virus, some time ago on either TWiV or TWIP, Dickson made an interesting comment about Japanese encephalitis virus transmission via worms of some kind. I think they might have been Trichuris or Ascaris. I can't remember. Could you elaborate a bit on this, please?
I bet he can.
Transmission between which species? Pig to human? What's the evidence for this? And do you have a reference? I'd be intrigued.
Oh, dear.
You're on the spot, Dickson.
I've said so much in so many ways. What I thought I mentioned was the co-infection of an animal with trichinella and Japanese B encephalitis. And trichinella prevented the infection of Japanese B encephalitis, as I recall, but the results are fuzzy for me. All I know is that somebody was working on both of these infections at the same time. Why they were doing that, I don't know, but they presented some interesting results, which I guess are worth going back and looking up. I don't think I mentioned Trichuris or Ascaris, but it was for another virus that I mentioned that one. That was for the swine influenza.
All right, searching PubMed with trichinella and Japanese encephalitis.
That's what I'm just doing right now, too.
Gives
Go for
us
it, guys.
5 papers.
Yeah, that's right.
And one of them is synergistic interaction of 2 agents in mice, Japanese B encephalitis virus and trichinella.
Right.
It's a synergism.
Who's the author on that one?
Lubinecki, Sipes, and Lucas.
Sipes. Raymond Sipes was the guy that was working on this.
That's
for a
the
long
paper?
time. Yeah, that was the one.
All right, there's no abstract. Okay, lovely.
Yeah, naturally.
There's another one, immunological sequelae of Trichinella spiralis infection in mice, effect on the antibody responses to sheep erythrocytes and Japanese B encephalitis virus.
Right. Again,
It talks about
probably.
suppression of the antibody response to Japanese B encephalitis virus
By trichinella.
by Trichinella.
Yeah, so in this one, basically, if they put trick first,
Yeah.
you get a worse Japanese B encephalitis. Yeah. increased
So
mortality. Yeah.
Is there a natural correlate of this? Why do
Well,
we care?
It's pigs. They're looking at pigs, I guess, as a natural vector for both of these infections, but that's not the case for Japanese bee encephalitis.
Well, they're here in these papers. They use mice, actually.
Yeah, but they were trying to relate this to some natural process where the claim was, and it's a good claim, that you're not just infected with one thing, you're infected with lots of different things. And what would be the effect if you put these two horrible things together at the same time? They got around several reviews on grants, and they got the grants for a while, but I think it petered out at the end because the model was
So
difficult.
his justification is that the virus, Japanese encephalitis, infects people,
right.
but it's rarely fatal. But sometimes it is, and why? He wants to know why.
Okay.
Is it maybe because you have trichinella?
Okay.
But that seems like a
That's a far-fetched
stretch, right?
That's a big stretch.
Yeah.
That's a big. The other virus that I mentioned a lot was the swine influenza and Ascaris. Ascaris eggs actually contain the virus, just as a physical carrier of it. It's they call it a paratenic type of situation.
Well, flu viruses grow in eggs, right?
Yep, they do. But I don't think they grow in Ascaris eggs. This was
right.
the one that Robert Shope Sr. worked on.
Okay.
At Rockefeller.
Lance continues, I'd like to recommend something to you all on the subject of vaccination, a book by a British family doctor called Mike Fitzpatrick, MMR and Autism, What Parents Need to Know. It's a great and easy to read review of the evidence or lack of it that MMR causes autism. What is particularly interesting is that Mike himself has a severely autistic son. He's a great guy. I met him because my wife did her family medicine training in the same practice. Your podcasts are keeping me going as I grapple with setting up a flavivirus immunology study in South India, which can be challenging at times. Anyway, please keep it up. I'm thoroughly hooked, and I really look forward to each week. TWiM and TWIP also. I'm slowly catching up with past episodes. Thank you, Lance.
Yeah, thank you.
Yeah.
Indeed.
Next one is from Ian, who writes, I was listening to TWiV 112 and heard Brent's letter asking about an old film he remembered watching in school that had animated white blood cells attacking viruses. I remember watching that same old film and was able to find it pretty quickly. YouTube never fails. And if it hasn't already been found, here's a link.
This thing was fun.
I particularly like how the viruses all meet up around a campfire at the beginning and conspiratorially develop their plan of attack, complete with evil cackling.
That's how it works.
Viruses
Evil cackling is the essence. What receptor molecule is that, by the way, Matt?
Campfire receptor.
The scavenger.
They're a pretty nasty bunch. Luckily, the white corpuscles have laser guns. Very good.
There is one inaccuracy in the film, though. It's around 5 minutes in. It switches to talking about the bacteria, and it lists some of the diseases they cause. And one of the diseases it lists is chicken pox.
Oops.
Little slip there, but otherwise a lot of fun.
Right.
My question is focused on the future of virology. I've always been interested in how scientific knowledge grows. It's always exciting to look at the history and see how it is rarely a linear accumulation of knowledge, but often a battle of competing theories and ideas with different schools of thought vying for followers and influence until eventually, over time, and with enough evidence, as an accepted theory arises. And then sometimes there's a radical breakthrough that reshapes everything. Do you think that in the next 50 years there will be such a radical breakthrough regarding virology, immunology, some sort of new technology or discovery that will challenge what we currently hold to be true and reshape the field? Thanks, as always. Sure. Yes, absolutely yes.
You think?
Yep.
Yeah, of
Sure.
course.
Everybody here agrees.
And if we knew what it was,
That's right, exactly.
then it wouldn't be a paradigm shift.
Quick to add.
I mean, there's all sorts of stuff we do now that we didn't have when I was a student.
I mean,
I would never have predicted it
just
either.
looking at a live mouse express proteins using these fluorescent protein probes is amazing.
Yep.
I remember the first time another kind of thing like that was done using selenium, radio-labeled selenium, to label the surface of a worm, a tiny little worm that was then put into the mouse. And wherever it was, if you made a two-dimensional mouse and did an X-ray of the mouse, you could find out where that mouse parasite was. But if you were to label it with a fluorescent protein, You could use the live mouse, and you wouldn't have to make a 2-dimensional mouse out of a 3-dimensional mouse. You could do it while it's alive and actually make a movie of this worm moving throughout its habitat.
Yeah, I remember when green fluorescent protein first came out, and it was just jaw-dropping. My God, you can actually do that, and it works?
Exactly.
Yeah, that's
well,
a-
the
that's a
atomic
big
force
change.
microscope is basically the same at that level, too, right? I mean, you can see patterns of benzene molecules. That's incredible. That's Kekule's dream. And you can see it right in front of you. That's
To me, one of the most amazing technological revolutions was DNA sequencing.
okay.
Because we used to sit around.
Sure.
I mean, I used to sit around in an RNA sequencing lab in the early 70s, and we would muse over how you could possibly sequence DNA. This is a good example of
It's
trying to look
easier
into
to
the
sequence
future and
RNA.
figure it out. And we're just clueless. And when the technology finally arrived, it was so elegantly simple.
Sure.
It's amazing.
My guess is that all of these things stem from a desire to get to the next level. I would like to look at a living cell inside the cell as if I were a molecule, and I could look around and see what the architecture looked like and watch the processes. There are these shows on TV called How It's Made and big giant factory stuff, and I'm fascinated by that. And the cell probably looks basically similar to that at some level. And wouldn't it be great to be able to do that? And so if you can express it as a thought, somebody will think of a way of actually doing it.
Ian's description of how science does progress, I think is quite insightful.
Yeah.
It is decidedly nonlinear.
Right. You betcha.
Ian was a student in my virology course this year.
Cool.
Yeah.
Good.
Nice.
But that's not how we write our papers up, is it? We write them linearly, but we don't think linearly.
Not at all. Yeah, and that is matter of fact, that, as Alan, I'm sure, will testify, is part of the trick. Taking that mess in your head and linearizing it.
Exactly.
Yes.
I thought discovery of mRNA was pretty cool. Remember who said that?
David Baltimore.
That's right. You weren't here, Dickson.
I was
You were in
not
India
here.
or something
I
like
was
that.
somewhere else. I'm
The
sorry.
next one's from Mitchell. He sends us a link to a story called Solar Energy Power Goes Viral.
Hey.
It's about making solar cells out of phages. And we'll put a link to that. And I'd like to tell everyone that Ian, the writer of the previous email. All right, so for my course, I asked my students for extra credit to write blog posts. So he wrote a blog post on this particular story, and it's up at Virology blog.
Cool.
So you can go check
What's
out.
Ian's major?
I don't know offhand. I
Good
have,
luck to Ian.
I have
Good
a
for
list.
him.
I have a list,
Yeah.
but I don't remember what his major is.
Okay.
All right, the next one is from Amit, and it's not Amit Kapoor, who we interviewed a few weeks ago. I'm pursuing research in immunology, working on immunological aspects of virus infection. I'm lucky to discover your podcasts recently. I've listened to some episodes and found it really interesting. You guys are amazing and make science real fun. I can only imagine the effort you guys must be putting in to make these episodes.
Weeks of preparation.
As I also came to know that there are TWIM and TWIP as well. I heard all of your TWIM episodes. Thanks for all the effort, you guys.
Cool.
I, though, want to add here that it will be icing on the cake for me and people working in immunology if you pick some papers with immunological aspects. You can add one infection immunologist to your group to discuss immunological aspects of microbial infections
Not a
as
bad
well.
idea.
It's just a suggestion, and not to say that I don't enjoy your podcasts in their present form.
So you need to start TWiV.
That's right.
Thanks for your efforts. Well, I think we could get an immunologist to join us.
Absolutely.
Sure.
Well,
Yeah, you've
that
had
would-
people that study vaccines.
Yeah.
And you've had innate immunologists, that's for sure.
The chairman happens to be one of those people.
Yeah, we could get someone. We can always add someone from time to time. We don't want to have an immunologist every episode. That would be
Antiviral.
antiviral.
That would be unlike ourselves.
That's right.
The next one is from Leo. Although I am an electrical engineer and my work is to develop cellular phones, I have been always interested in biochemistry, toxicology, and virology. Actually, I have listened to all your episodes, and you both make me think that in my next life I would study to be a virologist. One of my
both?
favorite. What's that?
Which both? There are four of us. Sorry.
Yeah, it's a good point. Maybe he's thinking of you and me, Dickson.
I don't know.
Maybe this is a really old letter.
That
don't
could
know.
be right. That could be
One
right.
of my favorite hobbies is to run simulations on the computer about protein folding. I recommend you to take a look to the project Folding at Home if you have not reviewed this recently. Yeah, I think it was a pick a long time ago. I'm writing you because I was looking for information about our tiny friend, the Tasmanian devil. After I watched a TV show about Australia, Tasmania, they commented about this kind of transmissible cancer which is killing the whole population of Tas devils.
Yeah.
Some reports consider that if not controlled, all the devils will be finished by 2030 or so. I would like to know if you may have some time to cover this disease in one of your future episodes. Do we know if its originator is a virus? Thank you very much in advance. Thanks for your achievements on divulgation of science.
You discussed this Tas Devil thing on TWiV at one point.
I think we did.
think we
We
did.
just briefly.
Yeah,
Yeah.
We did,
this sounds
we did,
familiar.
We did.
But briefly, this is it's a transmissible cancer.
Yes.
It's very interesting. It's
Well, I mean, they
it's
sort of inoculated
transmitted by probably by bites.
Exactly.
And these animals fighting with each other, and
Yeah.
so the bites are from the face to the face.
That's right.
And it grows to the point where it interferes with their eating and otherwise
Yeah.
makes them sick. And it's apparently there's a couple of other examples of this in nature, but fundamental to the transmissibility of the cancer is that apparently the Tasmanian devils represent a very restricted gene pool, so they one individual can't reject tissue from another individual very successfully, and that allows for the transmissibility of the cancer. And it's trashing them. It's not good.
Nope.
And I don't know anything about I don't think there's any evidence one way or another for a viral origin for this thing.
Not necessary.
And there doesn't really have to be.
No, that's right.
But we don't know what's the nature of the transmissibility, right? Do we
Well,
know?
I
It's
mean,
a
it's
bite.
contact.
A bite.
But we don't
That's
know.
bite.
We don't know.
We don't know what the agent is
the
that's
agent-
transferring it, or if it's just the tumor cells.
Probably just the tumor cells.
I
That
think
was
it's,
my understanding.
contamination of the teeth, and they would just bite the face of another one, pick up some of the tissue, and then turn around and bite one of their mates.
That was my impression.
Yeah, that's what this article says here. That it's a
nasty little
transmissible
critters.
cancer. Yeah, it's the cells itself that are
They are
being
nasty
transmitted.
critters.
And because they're so inbred, this
Yes.
this can happen.
The only
Right.
way they're going to survive is in zoos.
That's what I think about you, Dickson.
You do?
I just came into my head. I don't know why.
I don't get that at all, but everybody else laughed, so I guess they did get it.
I'll cut it out. I'll cut it out for you.
No!
No, don't do that.
I'm just kidding.
Don't do that.
I'm not making fun of you.
I'm fine with that.
Leo, by the way, works for RIM. Let's move on here from Dave. I came across this article on the possible future role of mathematics in virology.
Yeah,
dear.
we're trying to avoid that.
Lord.
It's called The Formula of Life in the New Statesman. Biology is undergoing a renaissance as scientists apply mathematical ideas to old theory. Welcome to the discipline of biomathematics. I try to reduce the amount of math that we have. Anyway, Dave continues, I've heard you and your panel members mention in past episodes that many viruses are icosahedral. However, I don't recall any discussion of the possibility of attacking viruses by modifying their geometry. Is the stuff on viruses mentioned in this article for real, or is this just wild speculation? I'd be interested in your thoughts coming from the perspective of a virologist.
Well, I read I had a look at this article, and actually their description of the math involved and the principles of the math involved in the icosahedral structure of viruses, I thought was right on the money and very interesting. And in fact, they don't really speculate very wildly at all, other than to say that if you can somehow disrupt the symmetry and therefore disrupt the assembly, you might have something for an antiviral. So if you had some compound that somehow interacted with one of the capsid proteins in a way that changed its interaction and would disrupt the symmetry and make the assembly impossible or proper assembly impossible, that could serve as an antiviral. I think that's perfectly reasonable.
Yeah.
So here is an interesting real life situation. So there are antivirals against picornaviruses that bind to the capsid, and they lock them so that they can't uncoat and release the genome. That's how they are antiviral. You can get resistant mutants, but the mutants are dominant negative inhibitors.
Ah, okay.
Because the mutations in the capsid that make them resistant to the drug screw up the capsids of other viruses that are assembling, and so they don't propagate very effectively.
Right.
So a single amino acid change in the one capsid protein will mess up the whole capsid, right? And a capsid is a perfect substrate because it's assembled from many subunits. So if you have just one among many that is bad, it can mess up the whole thing. So that's actually a validation of this idea also.
But you get a lot of incomplete viral formation naturally,
Sure,
don't you?
absolutely. Yeah. All right, let's do one more. And we'll do this for Rich. This is from Stephanie. So this is one of the letters from my virology class. In TWiV 124, Dr. McFadden says that poxviruses have the ability to steal genes from the host. In fact, this is precisely how they acquired the genes, similar to those for MHC class 1 and different cytokine receptors that were discussed in the first part of the podcast. However, if poxvirus doesn't need to go into the nucleus to replicate because it makes all of its own replication machinery, how and when does this pickup of cellular genes occur?
Okay, so I have two spins on this. First, as we've discussed before, I think this notion that the boundary between the nucleus and cytoplasm is absolutely black and white should be taken with a grain of salt. I mean, there's during the course of any infection, there may be some degradation of the nucleus. There can be DNA falling out here and there are opportunities for recombination between the virus and various cellular genes. So
Life is messy.
That's all theoretical. As a real practical example, I put in a link here to an article about foulpox virus. This is just a recent example of a phenomenon that's been recognized for some time, and I find it absolutely fascinating. This is a characterization of a pox virus. It's a pox virus of birds, foulpox virus, that has integrated into its genome a retrovirus. Okay, so now retroviruses, the retrovirus double-stranded DNA spends some time in the cytoplasm, correct?
It does. Yeah, well, in a complex of some sort, yeah.
Right. And so there is a real opportunity there for interaction between a double-stranded cDNA copy of a retroviral RNA genome, which is nice because now you've taken cellular genes and gotten rid of all of the splice sites and stuff like that, because you need a cDNA copy to be expressed in a poxvirus genome because there's no splicing. And recombine that into the poxvirus genome, and, with evolution, all of the other recognizable retroviral sequences are going to go away, and you wind up with a cellular gene or a cellular genome in the virus. So that provides a real life example of a mechanism for incorporation of cellular genes via a retroviral vector into the poxvirus.
Yep.
Wow. So this raises another question, though, to my mind, and that is when you teach at a certain level, because this is one of your students, Vince, this was an undergraduate course. Mm-hmm. And so what you're teaching them is the executive summary of the executive summary. Sure. So when you say that this virus replicates in the cytoplasm, you don't go into the nuance of, and sometimes it might get into the nucleus because there's no clear difference between the cytoplasm and the nucleus. Well, yeah, there's this nuclear membrane there. Nothing is black and white, and yet we teach.
Or black and yellow.
Well, in the case of hepatitis virus, we teach, though, in a way which makes everything seem ironclad, and there's no exceptions to what we just talked about, because there's no time to give you the tapestry of that particular bon mot, that little item that you're talking about. There's no depth to any of this, so that when a question like this arises, and then you have to give that answer, now that student's going to go back and say, how much else that I taught has validity at certain levels.
But that's the nature of science.
No.
Too.
I'm not saying it's bad. But it's like finding out, as a kid that, these are the do's and don'ts. But when you get older, that's the maybes and the maybe nots.
You make it.
Science is that way too. We have a set of knowledge that we're pretty sure about, and we find new things that say, hey, that's surprising, that's unusual, but we can it really kind of fits when you allow for the looseness in the system.
Exactly.
One of the things I like about this point you're making, Dickson, is that in my introductory lectures, when I'm teaching virology, one of the points that I always make is that virology is all about breaking the rules. Okay, you can come up with a couple.
Sure, that's right.
You can try and generalize.
No, you're
okay?
right.
The only real generality you can come up with is that they all have to use messenger RNA somewhere. Okay,
that's true.
But everything else is, the answer to every question is kind of, well, it depends.
But you've got a grandson now. What are you going to do about that? You'd like to raise your kids in a certain way. I mean, I don't think it applies to that situation, but we certainly treat it that way for most of our adult lives. And that's why we use the word, well, it's a surprise to me that worked like that. Why? Because you didn't hear it like that. When you heard it in class, it was totally different. So until you start to lay your hands on the research machinery and get involved in interpretation of data and conducting experimental work, it's very difficult to imagine any other way except black and white results. Right?
Yeah, and that's learning that it doesn't work that way is also part of understanding how nature works.
Right. Exactly.
DNA replicates and makes RNA, which makes proteins, and that's the direction it goes in. Except when proteins replicate and RNA replicates and makes DNA.
That's right.
So you make an important point, Vincent. This is something we all ought to keep in mind and incorporate into our teaching, is that it's not black and white.
No.
Right.
Especially in virology.
That's right.
Cool. No, because when you teach, you have to teach that way initially.
You do. There's no time.
Oh, there's no time, and it would be terribly confusing to say this is the way it is. Except
there's one guy that taught at a school that I went to that taught for 4 years. He taught history. All right. And he taught in the freshman year, the sophomore, junior, and senior years. He taught one course for each of these things. And he taught year 1 of French Revolutionary History in freshman class, year 2, second year of French Revolutionary History, year 3, third year of French Revolution. Year 4 was the last year of the French Revolution. And he went into all of the details. By the time you finished his 4 courses as a senior, you had one of the most comprehensive views of a revolution that's ever been
sounds great.
Talked about. It was amazing. It was amazing.
I'd love to take that course. I just finally downloaded this article you linked to, Rich. This Foulpox virus with the retrovirus in its genome. It's an Italian veterinary journal.
Yeah, right.
It's cool. It has an abstract in Italian.
Yeah.
Vincent,
you know how to say chicken, what is it, Foulpox virus in Italian?
I didn't try. Could you do it for
Virus del diftero vaiolo aviario.
There you go.
In polli da cortile non vaccinati.
All right.
I could go on. I love
I love it. That's good.
Okay, let's move to some picks.
Oh, dear, yes.
Boy.
Let's start with Matt. He has the longest pick on record.
Yes.
Well,
We
I
can
copied and
probably just
pasted,
link to this in the show notes, right?
Sure.
Right, I think the discussion could be equally as long, so if we can figure out how to say this concisely, that would be great. But I picked a petition to the NIH that was authored by Dr. Robert Ben-Ezra from Memorial Sloan Kettering Cancer Research Center in New York. This petition was signed by over 2,300 scientists, and at least 400 more wanted to sign it after they heard about it. So a lot of people agree with this petition, and it is arguing the fact that reducing the number of times an R01 application can be resubmitted to the NIH will prevent the outstanding projects from being funded and will actually especially hurt young investigators. So R01 grants are the foundation of scientific research in the United States. If a lab is to be successful, they pretty much need to get an R01 as their primary grant, at least initially. And prior to 2009, you were allowed to submit an R01 application 3 times. The first time it gets reviewed, you get some comments back. If it wasn't funded, you're allowed to respond to those comments and do it one more time even. In 2009, they actually chopped it down to only 1 resubmission. And maybe that doesn't sound so bad on the face, but Dr. Ben-Ezra's central premise that the review process is not perfect, and it is impossible to differentiate between great and very good applications. And since only a small number of these are actually being funded, there are many, many worthwhile proposals that are killed early because of this. And so I guess if you really think about this in more detail, you can think that each subsequent round of review would make an application better because you're responding to the esteemed colleagues' comments. And it also would provide, each additional round of review also provides greater resolution between how an application should be ranked. It's, obviously the best perfect application is going to be funded on the first try, but each additional round you take a closer look at the leftover applications, and hopefully then you can see what are really the worthwhile fundable proposals. Also, a resubmission rarely gets the same reviewers. And so that's a big problem, where you respond to somebody's comments, you put in a resubmission, and all of a sudden somebody else's criticisms are being applied to your grant. And if you get another shot at that resubmission, you're probably going to be less open to perceived weaknesses. So those are just reasons why resubmissions help make your proposal better. But the big problem is if your grant doesn't get funded, in this case in 2 submissions, NIH requires you to basically drop that line of investigation. You have to submit, any additional proposals you submit have to be new proposals. They don't tell us what a new proposal actually is, but they say it has to be greater than a certain percentage different, or it will actually be returned without being reviewed. And that is particularly difficult for young investigators to live up to. We start a lab on limited funds, and in a limited amount of time, we try to put together preliminary data to support a line of investigation. And if all of a sudden that line of investigation is shut down, we don't have other projects to turn to write new grants about. And so we're in a tough situation when it comes to putting together a new application. So I think the initial change, or the initial chopping of the third submission, was to reduce the time to funding, which, if you submit an application at NIH, you don't want to wait a year and a half to get it funded. And of course, this does reduce the time to funding. A grant that gets funded will be funded sooner. Young investigators that get an R01 will also get them sooner, but what they don't talk about is that when your resubmission is rejected and you have to write a new grant, that adds a ton of time to getting funded. All of a sudden, you're writing a new grant, hopefully, that is unlikely to be funded on its first submission, and your entire research program is basically just put on hold or put into jeopardy while you're doing this. So, this is basically a huge issue then for not just every scientist who's writing an R01, but particularly for new investigators. And this really prevents some of us from going forward and actually doing science, publishing, and planning new research directions because we're caught in this just getting that first grant process. The solutions to this, adding an extra round of review may not actually fix the problem. Maybe adding an extra round of review to resubmissions that had a certain percentile or were scored sufficiently high is one answer. So if you had a resubmitted R01 that got in the top 25% of grants, then maybe you should be allowed to have a third submission because it's almost there. And there is really very little difference between a 20th percentile grant and a 10 percentile grant. And so that basically everyone had already agreed it's a great idea. It's just not quite perfect yet, or it just by random didn't get a perfect score. And so to get one more chance on that grant just makes a ton of sense. The only real solution to this is more funding at the NIH. And there are possible ways to maybe spread the money out a little better between investigators. One of those is a hugely controversial idea of limiting the number of R01s any lab can have, or at least making it more difficult to get an R01 after you have one. Not to renew that R01, but to add a second R01 on top of it, or to limit the indirect costs an institution can take. Each institution negotiates a rate with the NIH that they can add to a grant and put in their operating budgets. And every school has a different indirect rate. And it's unclear why one school takes 50% off the top of a grant and another school would be able to take 100. And certainly limiting it to a reasonable rate would spread the money out a lot better. That will never happen, but it's still a good idea. It would be great if the NIH could actually tell us what a new application is so that we would know what rules we have to rewrite our applications to be applied to. It would be very bad if I spent months putting together a new R01, send it to the NIH, and they send it back immediately telling me it wasn't new. I would have, basically killed my lab if I had done that. Or do away with resubmissions completely. Make every R01 application a new application and let people continue to write about the same thing over and over again if they really want to. Of course, that makes the review process more arduous, but why should the NIH be able to tell investigators what they want to try to work on? The funding process is the big cut for that anyways. I went on for a long time there, and it's obvious.
No, but you articulated the whole thing very well, I must say. That was an excellent presentation.
Many levels. It hasn't changed since my day either, so I totally agree with everything you're saying. But have you ever served on a study section?
Of course not. I think I need to get a grant to qualify for that.
Okay, so once you have your grant and you get on that study section and you see the same grant coming back 5 or 6 times, which I did, everybody in the audience goes, oh. All right. Now that doesn't happen anymore, but it used to happen a lot.
Happens
and
at NSF.
It was
you can
tweaked
go back
and
over
tweaked
and over.
And tweaked. And you know what? At the end of those sessions, you know what we did? We said, to hell with it. Give them the grant.
Well,
come on, just give them the grant.
Persistence, I guess. But
that's right.
At the same time, I mean, I'm sorry that it's a hard process, but it should be an easier grant to review if you've seen it before.
Listen, I hear you. It is. It is.
It's also hard to write these grants.
So. My objection.
And none of this discussion would be happening if the pay lines were higher.
Right.
Absolutely.
Money would fix it immediately.
That was the most discouraging.
And even if I go ahead and get that R01, the next R01 is going to be even harder.
Yeah.
Well, money isn't forthcoming, that's for sure.
I used to say the only thing that was worse than not getting your grant was getting your grant.
At NSF, they allow multiple resubmissions, and the problem is that they have very little money. I was just talking to someone on a National Science Foundation study section, and they say you give a grant a wonderful score, and there's no money, so it comes back over and over and over again.
Exactly.
So money is the solution. And one of the solutions that has been suggested is if every university would pay half of the salary of the PIs, that would free up enough money. Yep. But they want us to recover all of our salary.
Yeah, I think that.
Because you are one of the major revenue sources.
You're the cash cow.
Absolutely.
You are the cash
cow. I think the institutions have become much too dependent on the NIH for their operating costs, and I think that they're going to have to pick up some of the tab here if we're going to address this.
Yeah, at Columbia, you may be second to real estate, but you're way up there as a revenue source.
Yeah,
it's true.
That's true. I know.
Well, I mean,
when I saw this come around, my biggest objection to it was that I do think that there's a mentality on study section, this queuing effect. Right. There's a mentality on study section when money is tight, that if there's 3 submissions possible, that the first submission they kind of go, they got 2 more chances.
Yeah.
And it doesn't necessarily get the same kind of review that it should. Now,
well, that's still going to happen with 2 submissions. It's just it's much more important in the second round.
It does turn the screws on the reviewers a little more, though. I think the mentality when there's just 2 submissions is I think different than the mentality when there's 3. Not that it should be that way. But I think it is. But Matt, I think you've articulated this whole thing very well, and you make persuasive arguments in support of this.
Well, the NIH response to this petition was not encouraging. They basically, and you can read that. I'm sure we can link to that on the show. But it's basically saying, no, our statistics back up that this is a good idea. Well, the statistics are a little misleading that they quote, and it even looks like some of the effects they're quoting occurred before they did this.
The NIH response focuses on this queuing effect as well.
Right. I mean, the time to award. And you know what? I'd rather take 3 cycles and wait an extra 4 months than have to have my R01 rejected and wait another 1.5 years, because most young lags cannot wait that 1.5 years.
Well, I think that one of the most important things is finding a mechanism to help out the new investigators. Okay, there have been several tried historically, and they help a little bit, but not enough. And I think some real special attention has to be paid to that.
Okay.
I agree.
Yep.
They're trying, but it's not good enough.
No.
Well, we could do a whole show on this,
We could.
unfortunately.
That'd be a
We
great
should
show.
do a whole show on this.
Well, let's talk about funding at some time.
We'll do
Dickson needs to leave. Let's do his
that.
pick.
I'm sorry that I have to rush, but I've got a 3:30 phone call. So my pick of the week is a book. The book is called The Big Thirst: The Secret Life and Turbulent Future of Water by Charles, get this one, Alan, Fishman. I know. I don't understand this. I just don't. But this is a very well written book. It was reviewed by Chemical and Engineering News, and it points up all of the problems that the world is facing with regards to the generation of drinking water and the despoiling of the environment at the same time. And I realize that doesn't directly relate to virology, but it indirectly certainly does, because there are lots of viral diseases that are transmitted by water, and or at least contaminated water. And we don't value water. It just says that throughout the world, we do not value the true value of fresh drinking water that's safe to drink. We don't put a price tag on it. We subsidize it. We use drinkable water to flush our urine and feces down the toilet. That's drinkable water. If you don't use
Not
that,
after you do that with it.
No, but before that, we could generate gray water for that purpose if we were clever. And there are clever schemes out there that being used where water is precious, like the Middle East. They do use gray water to flush away their black water. So it's just a very, very, I guess, poignant time in our history, because we're suffering from the United States is suffering from its largest flood ever at this very moment. And, Rich, you guys are suffering from a drought that's been going on for a long time. You draw a line at the Mason-Dixon line of the United States, and you've got floods in the north and droughts in the south. And
Tell me about it. I can't get my boat in the water.
Exactly. It points up all of the horrible weather changes that are occurring. It may not be climate change, but it certainly is odd weather out there. So at any rate, that's my pick of the week.
Thank you, Dixon.
You're welcome.
Goodbye.
Sorry
Thanks
I
for
have to
joining
run.
us.
Okay, guys. Take
See
care.
you, Dickson.
Take care.
Bye-bye.
All right, Rich. Let's have
All
your
righty.
pick.
I'm gonna claim 2 on the basis of timeliness here.
Sure.
But I'll be quick. First, I want to speak out in support of. I've been thinking about this, about doing this pick for a while. Data in support of the funding issue problem. I've got a link here to a page from the National Institutes of Allergy and Infectious Diseases, which funds a lot of the virology, most of the virology. That is an archive of final institute pay lines by fiscal year. And this is a chart that has fiscal year from 1999 all the way to 2010, and it lists the pay lines for RO1s, which Matt has nicely described as the primary funding mechanism for individual labs, and several other funding mechanisms that they get a different kind of score called an overall impact score. It doesn't matter, because you can see the trend in this. In the case of RO1s, it's done by percentile, which I think it's a complicated calculation, but it very roughly translates into the percent of grants that are funded. But importantly, what you can see is that whereas in the early 2000s, say 2001, 2002, 2003, we're talking 22nd percentile, there has been a steady decrease over the last decade to the point where the most recent pay line for established investigators in 2010 was the 11th percentile. So roughly speaking, you're getting half the grants funded than you were before, and that is not good. Those are the data.
Pretty sobering.
Yeah.
You know, one question I've had is that the percentile of total grants that were submitted, or is that the percentage of grants that were scored in that study section?
It's percentage of grants in that study section over the past 3 meetings of the study section. I think that's right?
Mm-hmm.
That were scored.
And it's not a percentage.
So that's actually half. Then that percentage should be cut in half, because only half of the grants get discussed and scored.
Right.
Good point.
So only 5.5% of submitted grants are being funded by the NIAID.
That's right.
It doesn't translate exactly into percent, but in a roughly, that's what's happening. The calculation is so complicated that I hate to put an absolute value on it, but I think the trend is indisputable, from a 22nd percentile down to the 11th percentile, and no matter how you cut it, it's not a large number. And think about it, I mean, you got to be in that. What's happening to the other 90%?
Well,
You
they
know?
have to submit a new grant after 2 rounds.
Right.
And that's a crazy thing that a brand new lab is competing against a lab that's been around for 20 or 30 years, building up expertise and data. We, as a new investigator, you get a little bump from the NIH. It's a few percentage points.
Right.
And so what basically happens is once you're graded or scored, your grant will be given a little advantage when it comes to funding.
You're graded on a small curve, essentially.
Really small curve. You're not going to get that again, so you better appreciate it the first time.
Yeah. Something has to be done to fix that, especially for new investigators, even with the limited funding.
Yeah, otherwise we're not going to have future investigators. Have very few.
Yeah, I hope I'm not pissing off the NIH too much in this. And,
Oh no,
not at all.
no. So my second pick is on a I guess a melancholy but somewhat lighter note in a way. Today was the last launch of the space shuttle, STS-135 Atlantis, and I watched it here on my computer in the morning. This is, Floridians have a special place in their heart for this thing, and there's a lot of people are going to be out of jobs. But at any rate, I've been a fan of the space program for my whole life, and I think the space shuttle program was really phenomenal, and it gives me chills every time I see this thing take off. I'm sorry to see the program go. I understand some of the reasons, but I hope it doesn't spell the end to space
I'm getting the
exploration.
soundtrack through here.
Sorry.
So I encourage everybody to have a look at this 2-minute video that shows the last launch and
So cool.
celebrate the success of that program.
Here, here.
I love when these go up.
Yeah.
But is any of that money going to go to R01s?
Not now.
Not now.
All right. Thank you, Rich.
Sure.
Alan.
Well, my pick is a book I read a while ago, and it just came to mind again recently. It's called Earth's First Steps by a guy named Jerry McDonald, and it's about the discovery of what is probably the best preserved fossil trackway ever discovered, or one of the best, from the Permian era. And the Permian era was 50 million years before the dinosaurs. And a trackway is actually, it was at one point a riverbank or another muddy area that animals walked across, and then through a combination of events that he talks about in the book, that became fossilized, and then subsequently was re-exposed, and now you can see these tracks of animals that walked on this very spot 100 million years ago.
So we're talking about several different tracks, several different animals?
Many, many different animals.
Okay.
And what he finds is this site in New Mexico, out in the hills of New Mexico, and he stumbles on it. This fellow had been a college dropout. He'd worked odd jobs and kind of hadn't figured out what to do with his life, but he'd always been fascinated by science and especially rock formations and fossils. And so in his spare time, he would go hiking around in the hills in New Mexico, and he was he decided to start looking for interesting fossils, and he found this trackway out there in the desert. And then the book is about that discovery and what he did subsequently, and the struggle that he had to go through to get this incredible fossil site preserved, because at the time there were quarries in the area that were digging this stuff up and selling it for flooring. And you can't replace this, because the events that create a trackway are very, very unusual. You have to get just the right set of climate and weather patterns to preserve these. As you can imagine, footprints, they tend to disappear very rapidly. But what's really cool about them as fossils is that these are records of the living organisms, whereas the other fossils that we have, mostly we have dead bodies. So with a trackway, you can actually tell things about how these in many cases, huge reptiles and amphibians, how they lived, how they walked, how they moved. And this is the discovery of one of the most significant finds in that area. And it's a really cool book. It's just a neat amateur scientist becoming a professional scientist and simultaneously making this incredible discovery.
So this is pre-dinosaurs. What type of animals are we talking about?
Well, there's actually one of the ones that he talks about extensively is often illustrated in it's iconically associated with dinosaurs. You know, the thing that has a big semicircular sail on its back?
Yeah.
The Dimetrodon.
Okay.
Yeah, sure.
That was not contemporary with Tyrannosaurus Rex and all the other celebrities.
Okay.
It was from the Permian era.
Okay.
And he rails about that a little bit. You know, this is not a dinosaur. But that particular lizard grew to be quite large, and not much was known about it. All that we had were some fossils of the, as I say, dead bodies. And one of the things that walked across this trackway, one of the many things that walked across this trackway, was several specimens of Dimetrodon, including some very large ones.
Cool.
So he's got footprints from it, and also a lot of other cool stuff.
So are they preserved there, or have they been wrecked?
Oh, they preserved most of the trackway. They actually managed to. He gets the Smithsonian involved finally. I mean, initially everybody just thinks he's a nut. Some guy wandering in the desert. He's not a scientist. He's just a geology student at the university and works odd jobs. But he eventually persuades folks at the Smithsonian, and he gets his senator on board with this, and even takes the senator out into the desert to show him the trackway.
Wow.
That's cool.
So
they got the site preserved.
Is this autobiographical, or is Jerry McDonald writing about another guy?
It's autobiographical. This is his work.
Okay, cool.
Nice. Great story. Thank you, Alan.
Yeah.
My pick is a lecture series which is called the Hertog Global Strategy Initiative. It happens every summer here at Columbia. And the lectures are live streamed and stored on Ustream, so I'll put a link for that. So this summer it's called Summer Fever 2011, and it included people such as Peggy Hamburg, the FDA commissioner, Robert Webster, who works on influenza, D.A. Henderson, the architect of smallpox eradication, Laurie Garrett, the writer, David Heyman, and I happen to be giving one next week.
All right.
Oh, yes.
Yeah, I was wondering who that was.
But
That's you, huh?
I'm in a different league. Tony Fauci. I don't know how I stumbled in here. But anyway, it's the other guys are cool to listen to. You can go check out their lectures.
Cool.
And that's
Right.
that. And in the interest of time, I will just say you can find a TWiV on iTunes and at twiv.tv. And do send us your questions and comments to twiv@twiv.tv. Matt Evans, thanks so much for joining us
It's great
again.
to be here.
Mount Sinai Medical Center here in New York City, Department of Microbiology.
Good to talk to you, Matt.
Oh, thank
Yeah.
you.
Yep, appreciate it. And I appreciate your coming up here.
Not a problem.
Alan Dove, thank you.
Always a pleasure.
alandove.com. Rich Condit, thank you, Rich.
Sure thing.
University of Florida at Gainesville. Dickson Despommier, of course, is at verticalfarm.com. And I'm Vincent Racaniello at virology.ws. You've been listening to This Week in Virology. Thanks for joining us, everyone. We'll be back next week. Another TWiV is viral.